Wonderful. Excuse me. Good morning, everybody. My name is Alex Klein, and as Dave just said, I'm the VP of Scientific Affairs at CurePSP. I'm with the foundation now for 2 years. My background is actually neuroscience. I'm a former neurobiologist working in a wet lab, working actually with animals trying to find new cures for, at the time, for Parkinson's and Huntington's disease. Now my switch to PSP, because all of these diseases are related to each other. And before I give you a little overview over the disease, I would like to introduce CurePSP, what we're doing, but also what the brain actually does when it's healthy. And I will give you a few fun facts. So, well, let's get started. As I just said, we start first with CurePSP 101. Then we have some Brain and Disease 101, our research activities, and I also would like to draw your attention to Ms. Oyler's talk right after my talk, and then we have Dr. Beauvais' talk here as well about Drug Development 101, or clinical trials, I should say. So who are we? We are actually a nonprofit organization founded in 1990. We're New York City based. We have only 9 employees. We're trying to do as much as we can with this little staff. And we're working in the prime of life neurodegeneration space or tauopathies. I will explain that in a second, what that actually means. And our vision is, of course, a world free of PSP, CBD, and related diseases. So what we do, we actually try to find a cure, try to help you. We provide care for families, but also, and this is really important, believe it or not, consciousness. It's still, in a way, it's a rare disease, but what we need to do is creating more awareness around you guys, but also around the healthcare professionals. It's really important. So what are the prime of life diseases? PSP, obviously, we have CBD here, and we have MSA. These are our core interest when we talk about diseases, brain diseases. But interestingly, there are 3 other diseases that are related to those 3, which are frontotemporal dementia, ALS, and CTE, the NFL disease you may have heard and read about in the media. All these diseases share actually something what we call protein misfolding. But again, I will explain that to you, what actually protein misfolding means. So how does care and consciousness look like? Well, we're here today. We're here for you to help, to understand, to share your worries, to listen to you, and to forward this also to our researchers and our pharma partners to really understand what you need right now. But obviously you need a cure, but that, you know, in that trajectory, we really need to come up with a solution for you, and this is what we're doing. You see, these are our patients, our friends, and then we do all this education. We have new materials out there right in the corner. Please feel free to take as much as you need. We also encourage you to take one for your neurologist. When you go home, go to your doctor, show him or her these materials, because healthcare professionals might not know enough to treat you. We also have Respite Funds. We all know the financial burden about these diseases and the care. Please check out our website, and you can learn more about what we offer actually to help you also financially. We have, again, this is dedicated to healthcare professionals. We have these, one-pagers that you literally can hand out to your doctor. And say, hey, this is not my language, this is your language, read it and try to make the best out of it, and this is what I have. That helps you also to translate in technical terms what you are suffering from. We also have YouTube videos. We recorded some lectures, all that everybody in the entire country can actually benefit from this knowledge. We also have now professionalized our peer supporter and support group training, just to give a level of quality and standardization across the nation. With the cure, I will come back after I introduced you to the Brain 101, and I talked a little bit about disease. But before we do that, let's do a little bit of fun stuff, because we're here because we all know that it's a bit of a heavy topic, I understand, but let's talk about the myth of the brain. And I want to break some of those myths, I guess, today. So I'm sure you have heard about that, that we use only a fraction of our brain. There is this number of 10%. And I cannot say it more often as, no, this is wrong. This is absolutely wrong. We use the entire brain. Because think about, what's very common is, for example, a stroke, right? And these are very small areas in the brain that are affected by a stroke. But if we would have 90% of our brain just lingering around, the chance of always having this profound effect of a stroke would be very unlikely. So it is not true. It's actually a misquote from Albert Einstein, who in a very humble interview said, well, boy, I think I'm not using enough of my brain. I feel like only 10% is working. That was then, as you now can understand, was taken away from the media, and then suddenly it was only 10% of the brain are active. Even if you go, use technology, you can actually prove that the entire brain is actually working. You can prove that by MRI or any other imaging technology. That's a funny one. Playing classical music to infants makes them smarter. Sorry, guys, no. I think it's based on a study that students were asked to solve mathematical questions or quizzes. And they were listening either to more distractive music or to classical music, piano music. And indeed, in that study, people with, listening to classical music could focus better. But actually, a funny story behind that is actually in 1998 in the state of Georgia, the governor gave out to every newborn a CD with classical music because he wanted to make Georgians smarter than everybody else in the country. But there are many studies out there right now that really do not support this. It helps you focusing, classical music. That's proven, but it doesn't make you smarter. Number 3, that's a tricky one. For many, many, many years, everybody thought that we cannot grow new cells in the brain. We have paraplegic patients, that spinal cord is still part of the brain. I will explain that later. So why are you paraplegic? Why not having new brain cells, new nerve cells, and fix the problem, right? So here's the thing. Technically, yes, we do grow new brain cells, but at a very low rate. So and then the rate is so low that let's forget about it in general, because otherwise, there are diseases out there, and if we could grow enough new cells, we wouldn't have disease, right? It makes sense. New cells are also born when we learn new things. That is something that really triggers also neuronal growth, but again, it's at a very low rate, and it's unfortunately insignificant for disease or for creating a new therapy. At the beginning, when people found out that we do grow new cells, the hope was actually that we can trigger this increase. But so far, it's not hasn't been successful. And with this one, I'm actually on a mission. It says, male brains are biologically better suited for math and science, and female brains for empathy. No, 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 no. But what I wrote there, and you may not be able to read it, is no. However, the power of social environment, i.e. our society, is very powerful, unfortunately. I'm going to tell you a really interesting experiment at the University of Ontario. They did that. So they had a group of people, male and female, asking to solve a mathematical quiz. To be honest, as expected, the male participants performed better. So, well, okay, maybe this is true, right? Funny enough, they had a new group coming in, and they told the new group of female participants, by the way, your brain works as good as the male brain in math. You know what the result was? Absolutely same results. So what I'm saying is, society is really dwelling on that fact that, Google Maps is a woman's best friend, so to speak, because they cannot orient, and all this kind of stuff. No, it's not true. Female brains can do exactly the same thing as male brains, and it's basically society that implies those things on us, and we learn this. And then also females learn like this. Males learn to really respond to those societal cues. So let's do a little bit of theory about the brain now. We learned about how it works and some fun facts. So when we go into the brain, you may have heard that from my accent. I'm European, I'm German, so maybe that's how my brain ticks. For us it's football, by the way, not soccer, but anyway, it's soccer. It's the soccer brain, right? But then, how does the American brain look like? Well, maybe that's more your brain, right? I don't know. Brett Bouvet can disapprove me later on. I don't know what he finds in his imaging studies. So here we are. That's the brain, right? So that's this kind of wrinkly stuff that you see when you look at the brain. And we really need to understand what's in there. How does pathology, how does disease work, actually? And so the main thing in the brain is actually a nerve cell or neuron, how we call it. And the neuron is really conveying message from one cell to the other and makes me, if I want to move my arm up and down, the nerve cells have to generate this impulse and really have to translate this to my muscles so I actually really lift my left arm here. So that's what these guys are doing. These guys are also storing in a network, storing memories and things like that. And I want to give you a few mind-boggling numbers, which I don't get. I really do not. And I have to be honest with you, I have no concept what 100 billion actually really means. But let's go through this a little bit. Well, we have 100 billion neurons in the brain. And this is one neuron, and the measurement, science is metric, so we have a micrometer. And when we take an average width of a neuron, this part here where you see these arrows here, this is 10 micrometers. If we then go up to 10 neurons, 100 neurons, 1,000 neurons, 100,000 neurons, and we all align them next to each other, this is called the nucleus. If we have nucleus next to nucleus next to nucleus. We're already at 3.5 feet, but we're only at 100,000 neurons. If we go to 1 million, we're already at 0.6 of a mile. Go to 1 billion, we're at 621 miles. And if we go to 100 billion, we have 62,000 miles of neurons in all of your brains. Everybody of us would have 62,000 miles of neuron after neuron after neuron. Just a reminder, Earth's circumference is roughly 25,000 miles. That is, I'm almost beyond my understanding. And then that also, why I'm telling you this is also, I would like to sort of hope for your patience that researchers still haven't really found a cure, because this is very complicated. Imagine, and these cells are now all talking to each other. How the heck are we ever going to find out how this works, right? Also, just to visualize this, we're based in New York. If you start driving to Chicago, here to the Bean, this is 793 miles. And this is 78 road trips, right? That's how long, that's all crammed in here in one brain. Or another comparison, I like that one as well, it's halfway up to the moon. If we align all these neurons one after the other, right? Because we have cells up here, they need to communicate through my body. So they're quite long. They're maybe 2 or 3 feet long, right? Those extensions. And if we, again, put that one after the other, we would go all the way halfway up to the moon. And again, this is all folded into our brain and has to sit there and has to work, has to function, has to have the right nutrition, has to write the correct input. And if anything goes wrong, you can imagine that it's actually quite easy to have something that really then gets out of hand, that we might not even be able to control anymore. Just to make it even more complicated, those neurons, those nerve cells that help you to communicate, to carry out behavior, need support. And those supporting cells are called glial cells. And by the way, we have 5 trillion of those. They're also sitting up there, all in here. So just to give you a gross structure of the brain and how it looks like, is you see here we have the central nervous system. It's part here, the brain, as you know, and then it goes all the way down here in the spinal cord. This is part of this, what we call the central nervous system, or CNS. You may have heard that before. And then we have all the other nerves that connect to the muscles here in the periphery, we call peripheral nervous system. There are two separate nerval systems, and what we are talking about most is actually, and in the context of PSP, CBD, and MSA, is actually the central nervous system. What do? The central nervous system here is, again, it's the brain, it's the spinal cord, goes down the vertebrae and the spinal column, and the peripheral nervous system is, again, anything outside of that CNS. Also, interestingly, the brain can be separated into functional areas. And you see, I mean, it's most probably quite hard to read from the back that we have motor areas, we have speech, we have vision here, we have language, we have posture and balance. And this is really important because we know if disease happens, for example, in the more motor area up here in the cortex, in the outer layer of the brain, you will have motor impairments, motor symptoms, right? If you have more frontal here, what we call frontal, and is concentrating thinking, behavior, mood, personality, that's all located in there. Of course, then you have more symptoms that are less motor, but more affecting those type of behavior. That's really important to understand. Another fact, we're still at Brain 101, is that you may have wondered that, elephants, whales, they have these gigantic brains if you see them, right? And for example, an elephant brain is just over 4 kilograms, which is, I guess, around 8 pounds. And then you have the marmoset here, the rhesus monkey, gorilla, chimpanzee, and a human brain at roughly 1.5 kilograms. Or 3 pounds. And you would wonder, well, why isn't the elephant the crown of evolution? Why aren't elephants running around and doing all the things we do? Why can't they talk? Why can't they just do things, right? And the interesting answer of this is, you just saw this scheme with the different functional areas, right? If you think of that, and if you just look at the number of neurons that are in these areas, and this is actually part of that makes us really human, I would say, is that the number of neurons in the cortex here in billions is 16.3 billions in humans, but only 5.6 in an elephant. So that's really makes a big difference, right? So it's not that the sheer mass actually matters. No, it's about the quality, about the number of neurons that can actually talk to each other in the brain. That makes us actually human then. So let's talk a bit about disease. What's going on in PSP, CBD, and MSA? When we talk about disease, we also need to understand, and I'm sure you will hear about this also more by Dr. Bouvet, but you need to understand what's going on, because now we know how a healthy brain looks like, but then we need to understand, well, okay, we have Homer Simpson up here. I hope we'll never have that type of brain. But then we have, you all have seen those pictures most probably when you went to your doctors, and these are so-called MRI images, and so you look into the brain and you can actually see what's going on in terms of, volume and things like that. And Well, you can see black. Black is bad. Pure black, because that means a sort of liquid-filled cavity. So you would assume that here is not a lot of black, here is a little bit more, and of course that sound looks really, there's very little white, so I guess this is a really seriously affected brain. And then if you look at that, actually that's why I'm bringing up these two healthy control brains here, because we really have to understand how to interpret those images. And that's actually one of the big problems still that we need to overcome in PSP, that imaging so far is good, we know how to use it, but it still needs some serious improvement. But as you can see, this is actually an FTD brain here, this is a normal brain, and this is my brain, by the way. You know, when you're a researcher and you're next to an imaging center and they need control persons, you have no chance not to say no. And actually, I was in that scanner for 1.5 hours, and at some point I said, well, I want to go out now. So, as I said, those 6 diseases, we call them prime of life because they strike earlier than Parkinson's disease, or tend to strike earlier, and they really, you know, hit you when the kids are just fine, they're out of college even, maybe have their first job, you stop paying them, awesome, you know, you have more money for yourself, and then, you know, boom, we have a prime of life disease. We have one of those diseases, and I think, unfortunately, you can relate to that. What's the shared mechanism? We call this protein misfolding. You know, we're built of proteins, and proteins have a certain structure, and this structure needs to be manipulated in a certain way so that they can work and function in the cells. And if anything goes wrong there, then we have a misfolded protein. And this is actually what's happening in PSP, CBD, and MSA. Different proteins, though, between those 3 diseases, but this is the unifying common denominator between those diseases. So what does it mean? For PSP and CBD, it's a protein called tau. For MSA, it's a protein called alpha-synuclein. Interestingly, by the way, tau is also misfolding in Alzheimer's disease. Alpha-synuclein is misfolding in Parkinson's disease. These are our big sort of friends, if you will. And that really cranks up also the number of patients. If you look at, we call, you know, any disease that involves tau as a disease-causing, you know, ingredient, so to speak, we call them tauopathies. So Alzheimer's disease is actually a tauopathy. And that also is actually the big hope we would like to bring across here right now, that we believe that PSP can serve as a model disease for finding a cure also for Alzheimer's. And you can understand that suddenly we have 5.5 million patients in the US alone. And that really is absolutely attractive also to pharma companies. Because, you know, we have 20,000 patients right now in the US with PSP. It's still technically a rare disease. It's also financially not so attractive to find a cure for those diseases. But, you know, that's actually what we believe and hope that we can help with the orphan drug status, that we are actually at the center of this development also for other diseases, such as Alzheimer's and Parkinson's disease. What happens? You see here, this is very small, I apologize for that. You see here, this is a nerve cell. And it travels all the way down here. And you see something, what I call is sort of speak a skeleton of the cell, or scaffolding. It really keeps the cell together. You know, these very long extensions halfway to the moon, you know, you have to stabilize that somehow. And tau is basically one of the bolts that really keeps this scaffolding together. And that's one of the main functions of tau. So in disease, what happens is when tau becomes misfolded, it cannot serve as a bolt anymore. It doesn't fit into the scaffolding. And even worse, it starts accumulating. It's not just hanging around there and not doing anything. It's actually starting to accumulate in aggregates. Those aggregates are actually even worse because they they do cause more aggregation. So it's a self-increasing process which ultimately leads to neuronal death, that this cell here will die, and then you have the atrophy in the brain, that's what you saw. I just have to watch the time a little bit. So, okay. So, how do we know then, or what are the differences between those diseases? Why does an MSA patient have different symptoms than a PSP patient? What I would like to show you very briefly here, you see sections through the brain. And you see a color code here. And you see that areas affected in MSA and PSP are different. And then remember that, I will show it in a second again, but remember that brain where I showed you the functional areas, right? So if I have disease right up here, might be actually more motor, and here, or more frontal, I should say, and here this might be even more motor in terms of balance, etc., which is a big problem in MSA. So I just would like to make sure that you understand that disease, I mean, even though there is the same protein sometimes between PSP and CBD, for example, or MSA is alpha-synuclein, different areas of the brain are affected. And then you see here again, this guy, different functional areas will be affected and will have, as a cause, will have different symptoms. So the cure, what is CurePSP doing to help cure? We have a venture grant program, we have special initiative grants, we have conference sponsoring, we have a genetics consortium, we work with the Mayo Clinic very closely together. We have Trish Karana and the PEP, the patient engagement program. We try to be involved in clinical trials, and we will soon have CurePSP centers of care. What are our venture grants? We have so far supported more than 170 researchers worldwide. We're not geographically restrained to the US. We want the best people. We have a scientific advisory board that picks the best applications to CurePSP. It's an NIH-like vetting process. We take quite some pride in this because we're a small foundation, and over the last 20 years or 25 years we have raised more than 30 million in supporting researchers. And that everybody can apply. It's non-profits can apply, academics, but also even small biotechs can apply, and we can help to bring a new compound forward. Then we have the PSP Genetics Consortium. We're very proud of that. Very recently launched by, again, you see here, we work with the Mayo Clinic. We have people from UCLA, Penn Medicine, but also from our European friends. We try to investigate on a large scale, how do we find any risk factors in our genome? We know it's a sporadic disease, it's a non-genetic, non-familial disease, PSP, but we need to find solutions for maybe there is more in our genome that we can identify also as new drug targets. We also work again with the Mayo Clinic, but also recently on that with the Tau Consortium, together with the Mayo Clinic Brain Bank. Since many years we do this. This is absolutely fantastic. We encourage you to start thinking about this if you are interested in. Researchers really need access to human tissue. We had quite a few failed clinical trials in other areas, but also unfortunately in the PSP domain. And I think it's one of the most important research projects actually we have is supporting those brain banks that we have a lot human tissue accessible for researchers so that they can try their new compounds on human tissue. This is very important. We do support families because we know that we acknowledge that you have financial constraints during the course of the disease, that we reimburse you up to $750 with, for the costs of a brain donation. Also, really soon, we hope to launch CurePSP Centers of Care. These are centers, self-reported, that are familiar with PSP. We will extend that to CBD and MSA. So what we hope is that we can not guarantee a certain standard of care, but at least we can gather information of centers. And it doesn't have to be a big center. I'm not talking about the big universities here right now. It's a local center where a physician, a nurse who has ever heard about PSP and knows how to treat and deal with a PSP patient. That'll be absolutely fantastic, and we do this and we hope to roll it out by October this year. Very briefly, I would like to also to tell you more about drug development and a little bit about clinical trials 101. Because I hear, every day I take phone calls. I hear the frustration from you, and right so. Where is the treatment? Where is the treatment? Why does it take so long? And with all the research going on, it's actually very, very expensive. But then also because of the regular way how science works, it's also a very timely process. I just wanted to kind of set the stage for Dr. Beauvais, just to give you some tools, some terminology that you really understand what we're talking about. We have 3 main phases of clinical trials. And I put the other phases in there because you will hear of them. And they're not really frequently used, and people use them differently, and it's a bit of a mess of how people use these terms. But those 3 are actually clear and everybody uses it. So let's go from the top. It's called preclinical screening or toxicity. So we have non-animal studies, we have animal studies. That's where the nerdy basic scientists are and look for new compounds. Then we have a phase 0, which is basically almost the same thing. It's more exploratory. People really use those terms interchangeably. But then the first important phase comes in here. First in human, dose finding and safety. This is also often with healthy controls. Then we have the phase 2, the proof of concept, and phase 3, the proof of efficiency. And then, of course, we have also, because we need to monitor, once a drug is on the market, we need to monitor the effects, because then suddenly we have a much larger population who will take this drug, and we need to still approve and closely monitor the effects of a new drug. I also would like to guide you through this very briefly. This is basically here from basic research to the post-approval period. And just to enlarge that a little bit, let's start with the drug development here, or drug discovery, it's called. We have basic research, we have the drug discovery here, and we have the preclinical. So what it means is with the basic research, it's the really, really, really basic research, kind of looking at what's happening there. Let's study this disease without really a targeted goal where to go. Once you have identified a few compounds that might look interesting, you go over to the drug discovery period. That's also when the chemists come in and start to modify a compound for better penetrability into the brain, for just improving the function of a compound. And then we also go into the preclinical, which is when we really start looking into safety and toxicity of those new compounds. And that's also when animal testing is taking place. And then we go to the clinical trials, which will hopefully end with an FDA-approved drug. So if you look at that, this is the green period here. Once you are confident that your new compound is actually something that you believe in wholeheartedly that will help a patient, you have to apply to the FDA. And once your application has passed through, you go for clinical trials. And we had these clinical phase 1, 2, 3. And at the very end, the FDA will review. You have to present all the results to the FDA, and they will review your success by an independent committee. and hopefully in the end it will actually be with an FDA approval so that the drug can be sold. Clinical trials are needed just as a summary, are needed to make sure that a drug works. That's basically the simple idea of a clinical trial. We encourage you to really be part of that. It's the only way forward that we can actually test for new drugs and the efficacy. We have those 3 clinical trials. Trials need to be randomized drug versus placebo. Otherwise, if you anticipate an improvement, and that's what then the placebo has to come in, you may actually feel better only by the fact that you're enrolled in a clinical trial and that you think you get a new drug and you really you collect the last resorts in your body, you really the full energy, and you really feel better the next time you be screened by your neurologist. So we have to make sure that you don't know what you're taking, either a sugar pill or the real pill. Because everybody who is participating in a clinical trial is highly motivated. We have to really make sure that it's not the ingrained motivation that keeps you improving, and it's only a honeymoon effect, by the way, it will peter off, or if it's a real drug effect. We have to measure all of this, and that's actually also a technical term you may have heard from your doctors when you read about this. It's called outcome measures. What are outcome measures as well? We have rating scales. We need to see if your motor behavior improves or not, if your cognitive function improves. We have some more unbiased measurements, such as biomarkers, we call them. These are biofluids, MRI, the whole imaging of your brain. If an independent doctor can analyze your images and say, yeah, you know what? There is actually more white. You remember the first images? There is less black, we see, or at least it stabilizes, right? There's no progression in the disease. And so I also would like to mention that clinical trials are extremely expensive. And that's why we need Big Pharma to help us with that. And what we have here, I just wanted to mention this, we don't have to go through the whole list, but these are all people who work on a clinical trial. Well, it's you, but then we have the investigators, physicians, scientists, the regulators, we have the companies, nurses, study coordinators, we have then independent raters and testers. We have, this whole thing here, the outcome measures, the rating scales. Also, we have laboratories who need to analyze your blood, everything. And that costs time and money. So, again, what happens during a clinical trial? We have the recruitment, we have a pre-screening phase where we'll discuss if you are actually eligible. Then once you're eligible, we need you to consent to take the drug and to agree with whatever is going to be tested in this clinical trial. And we have a screening visit, and even then, we still need to figure out if you are actually the correct and the right patient for this clinical study. And then, of course, we just go through the whole process with phase 1, 2, 3, or whatever part you are in, and then we hopefully have the FDA approval. So just again to repeat that, after the FDA approval, we have the post-approval research, just monitoring very closely together with the regulators and pharma if the drug is safe and doesn't cause any unwanted side effects. And this is maybe a bit the frustration picture for you to see how much. Well, this is a study by Tufts University. It's 2.6 billion. There is some criticism around this study saying that it was way too high because non-profit and governmental organization actually contribute significantly to this process. reducing the numbers, but if you go right into the private sector, I think that number might actually be quite correct. And then here, 10 years. That's an average, and I think that's already a very ambitious time period. But this is peer-reviewed, this is published, so that's, I take this now as my numbers. And with that, I would like to thank you for your attention. All right, and I would like to introduce Sarah Oyler from NYU Langone. We're very excited to have her here. CurePSP has funded, last year we started, a project where, you know, it's actually really outside the box project. I'm very proud of this. These guys actually go to the patient rather than you go to the doctor. And I think Sarah will tell you more about it, and it's very exciting, and it's an in-home visit program, and really have fun listening to her.
All right, thank you. My name is Sarah Oyler, and I'm from NYU. I'm going to speak a little bit about a study that we're currently engaged in called CARE-PSP, Understanding the Needs of People with PSP and Related Disorders. Hopefully this will take no more than 10 minutes of your time. Okay, so who are we? These are just pictures of people on our research team. We're a group of movement disorders specialists led by Dr. Jory Fleischer at NYU, and we're interested in understanding the needs of people who are living with PSP, MSA, and CBS, and related disorders. What are we doing? We're conducting a study to identify how people with PSP and related disorders use and seek health care. We want to know what kinds of care is helpful and what needs may not be met. We're interested in understanding the quality of life of patients and caregivers, and how quality of life changes over the course of 1 year. Why are we doing it? People living with PSP and related disorders are often grouped under the umbrella of Parkinson's disease, but we know that they and their care partners require many different types of care, and we hope to identify unmet need and use that information that we acquire to help guide future studies. Potential impact of our study is to guide future studies, use this as a model of care to improve quality of life and care partner strain for individuals living with PSP and related disorders, and improve future care. So who can participate? You can participate in the study if you've been diagnosed with PSP, MSA, CBS, or an atypical Parkinsonism. You're over 40 years of age, and you voluntarily agree to participate. But not only patients can participate, but also their care partners. It's very easy to participate. You'd need an internet connection and a computer, 30 to 40 minutes. And if you'd like more information, you can either share your email with us today, and I have an email sign-up sheet by the materials table, or you can call Naomi Fried, our research coordinator. Her number is here. We also have a flyer you may have received, and if not, I can make sure that you do receive that so that you can call to receive more information. And that is our lovely research coordinator who you would be speaking with. So accessing the survey, if you provide us with your email address today, you can expect an email with your personal link to our survey in your email in about a week. You can also write down this particular website and just go to the survey yourself, or you can, of course, call Naomi Fried and she will provide you with the information. We wanted to briefly go over some of the things you will see when you open the survey. So all research requires approval by the Institutional Review Board or an IRB to make sure that the research participants are fully aware and informed of their rights and any risks or benefits of participation. So when you first open the survey, the first page you will see will be a whole bunch of text, like this exact slide I'm showing you right here. And at the bottom of the text, you'll be able to click yes or no to agree to participate. If you agree to participate, if you click yes, you'll be directed to this page. And again, because the IRB wants to ensure that all people who participate are fully informed, the second page will just be a list of 4 questions. None of them are meant to be trick questions. They're just to make sure that you are fully aware and understand the purpose of this survey and this study. This is just a screenshot of what some of the questions are going to look like. We tried to make the buttons as large as possible and as easy to understand as possible, but that means that some of these answers are not in the correct order. So make sure that you read through very carefully to make sure you're answering the way that you want to be answering. This is one additional screenshot we wanted to make sure we pointed out to you. There's going to be lots of different types of questions on this, and this one in particular has a slide bar that you'll have to slide. It'll be just that blue box in the middle asking you today, what is your health status, the worst health you can imagine or the best health you can imagine. And then once you slide that little blue box, a number will come up in that little white box that will correspond with your answer, just so you know. After the survey, so 1 year after completing this survey, you'll be contacted by email to conduct a follow-up survey. We really hope that you will complete this survey, the follow-up survey, because that's really the only way that we can measure your quality of life after the course of a year. If you have any questions about this, you can contact Dr. Fleischer's research team at that number, or like I previously mentioned, you can provide us with this email, with your email address, and we will email you in about a week. So that was my very quick spiel on our study that we're doing. We're very thankful to CurePSP for helping us to complete this study. If you have any questions, please let me know.
So we have time for a little bit of Q&A. If you have questions right now for us, yeah, we're here. We're around all day. You can grab us and we sit down and yeah, so floor is up to you. So if you have questions right now, yes, please. We have a mic.
Be done. What is the connection? Because that's an invasive procedure. I want to know why so many of the clinical trials ask for both volunteers and patients to have a spinal tap.
Yeah. So the answer to that is, so the question was, I don't know if everybody really could listen to that at the beginning. So why do we have CSF, cerebral spinal fluid taps quite often in clinical trials? It's an invasive procedure. It's not very nice. You know, I'm suffering actually from sciatica. I had 2 injections the last 2 or 3 weeks. It's not nice. I hear you. I hear you. And then the worry is, somebody is poking into your spinal cord, and you have these ideas. Am I going to be paralyzed afterwards? What's going on? And so it's a very safe procedure. First off. Second off, it's a bit uncomfortable. But the only way you can actually look into your brain, without technology, if you really need the composition of what is in the liquid that is surrounding your brain, the only way of getting into that is actually by tapping into your spinal cord. That's the safest place to really get a sample from CSF. So that's why, and it's the analysis of that is really important to see if a drug works or not, or what is actually happening in the brain. You can also measure inflammatory markers. Anything that actually goes on in the brain can be tested by that. And unfortunately, so far, it's only by this invasive procedure. I wish we could have. I mean, we're trying to have better analysis of biomarkers by drawing blood. looking at that, but it's still early on, and there is a lot going on, particularly with blood biomarkers, but I think right now, as we are, we still need CSF. Yes, please. All right. Sorry.
Yeah, I've got a question. In the handout, you grouped CBD and PSP in one of the slides sort of together. Could you explain the similarities and differences between those two?
Oh, wow, do you have another hour? That's a difficult question. There are, yes, you can distinguish between them, absolutely. And symptomatology is also different, right? CBD is a bit more frontal, less motor symptoms. Also in the brain, different areas are affected. Interestingly, it's the same protein that misfolds. So that's why I grouped them together. And generally, they are actually part of the same family of diseases. I cannot tell you what triggers one or the other. I think nobody can so far. Why somebody has PSP and the other patient has CBD, that we cannot tell you because we don't really know what triggers, what's the cause of these diseases. Right now I can only tell you that different areas of the brain are involved, causing different symptoms. And that's how doctors really diagnose them. It is very, very complicated to really put a final diagnose on both of those type of patients. What we have, and that's why we're working with Mayo Clinic together, is what we encourage people is to donate the brain so that the family in the end will receive an autopsy report. And only the pathologist at Mayo Clinic can actually tell us definitely that according to where tau actually accumulates, this is CBD or this is PSP. And we can talk about the rest at length later.
So I actually have two questions. One is, when you mentioned the Center of Care, can you elaborate a little bit more on that? And is it anticipated that it would be specialists or primary care physicians, and also what is the goal? And would there be some sort of like training or certification, or is it just identifying somebody? And then the second is, do you have any numbers, or did that study indicating the cost of bringing a new drug in the U.S., did that look at other countries or other places around the world to compare? And is it more like an FDA or a pharma, in terms of the cost? I'm just, did that compare anything to Europe or other places?
Yeah, I can do that. So first question, with regard to the CurePSP Centers of Care, we're super excited. And I can tell you that initially we thought we can actually pull off some sort of center of excellence network as we see in Parkinson's disease. The problem is that in Parkinson's disease there is just simply more funds available. And those centers will then again, what you said, you need training, you need grants, you need study coordinators, center coordinators, suddenly this becomes a multi-million project. So we knew that we cannot pull this off in the field of PSP. But what we wanted is, as part of education and awareness, what we can do is actually we will have twice a year, we will have an application deadline where centers, whoever believes is familiar with PSP, can apply. We have a survey monkey, you can respond, it's self-reported, and then we will grant you as a center a plaque on the wall, basically. But what we then also can put on our website is a map where our patients can go and identify regionally where is a center that knows of PSP. And it doesn't have to be a center of excellence. It doesn't have to be a center that has 5 MRI machines in there. It's actually, it can even be a PT/OT office that has seen 2 or 3 or 10 PSP patients. But at least we know that once the patient comes back from the major center, that we have some, not control, but we have some good care with people. Because when we have the majority of our people, once they go into assisted living or hospice, for example. These institutions are completely clueless about what actually a PSP patient looks like and how to treat and deal with them on a day-to-day basis. So this idea was then also with the self-reported survey and center of care was actually originates from our friends from Germany, from the PSP Society of Germany. They have a beautiful clinical guide, and they started now 3 years ago with 40 centers across Germany. And mind you, Germany is only 2.5 times the size of the state of New York. It's tiny, and they had already 40 centers. The second edition has already 80 centers. And again, these are not the major universities, these are not the major centers, but these are regional hospitals who have some idea of PSP. And then what the beauty is of this is that you can now then start comparing those centers as well with services. We will make them use exactly the same language. When we have those, centers on our map, and you hover over a pin, and then a pop-up will come, and it will show you what this center can actually offer. And with regard to the numbers for clinical trials, that's one of the big. This was a pharma-sponsored study, but it ran through Tufts University. For that reason, I think, and that includes all numbers, FDA approval, post-approval. That's kind of what, that's what pharma has to scribble in for a new drug. Again, there was some criticism at the beginning, but these guys were out there and they really defended themselves, and I think the numbers are on the high end of the spectrum for sure. And I think this might be even only a mean, because I think as soon as NIH kicks in for a long period of time, maybe that number is reduced. But this is, for me, still a number I can accept and wholeheartedly present to you.
Hi. In all of the research that you've looked at, have you seen any connection between head trauma early in life and onset of PSP?
I have to think about that a bit, how to phrase this. I think what you're referring to is also what we call CTE, chronic traumatic encephalopathy, which has been identified actually quite a long time ago now, also in boxers, in any people who do impact sports. We know that anybody who suffers from concussions may be exposed to a higher risk. That is true. But if you talk to my friend Dr. Robert Stern from Boston University, he will say that even that we're talking about concussion in this context is already propaganda by the NFL, because it's not the individual concussion that you should be worried about. If you have enough time to recover and you give yourself time enough to sort of rest during that period, and then I think the individual concussion is not really something you should be worried about. It's more the repetitive subconcussive impact. And that's actually when we start getting worried about our college students and other athletes. It's the repetition that is bad for your brain. Actually, there was even a New York Times article out there last year looking at synchronized swimming, because, when they meet underwater, the kick is basically a cue. It's not that they're all fighting underwater. It's literally that's what they need to come back up. And so there is actually one case already of a former synchronized swimmer who suffers from CTE. So I think the answer is linked to PSP. I don't know. I don't think so right now, to be honest. But to be honest, this is still early on. We don't know. And if it would be CTE and not PSP. All right, so who's next?
I was just wondering, I know it affects the senses, the seeing and you're swallowing and throat. Has there been any studies or anything on that it affects the hearing of people with PSP, MSA, or CBD?
Not that I'm aware of, to be honest.
Was it easy done? You outlined the approximate 10-year timeline up till FDA approval of a drug. Is there anything in the works right now for PSP, and where in the stages might that be?
Oh, you can ask our friends from BMS Biogen and AbbVie, who are in the room here. There are very promising clinical trials out there right now. And right now what we're looking at that's the most promising or closest, I guess, to be promising is antibody or immune therapy. But again, it's still in an experimental phase. It's clinical trials. You can also go to our friends here at the front. AbbVie is having a booth here. But then, there is a lot of hope out there. There's a lot of approaches from different angles as well, because we need to make sure that we don't focus on one therapy only, because the silver bullet is quite unlikely to happen. But what we have, there's a lot of things going out, like repurposing of drugs, of clearing the accumulated tau, right? So you have the tau in the cell, and we do have garbage disposal system in the cell. And that works pretty fine. But in PSP, this is reduced, and we have a toxic load of tau. So it's kind of a catch-22, right? So if we can develop drugs that increase, for example, the disposal of toxic tau, and then at the same time we have one of the antibodies that as soon as the tau leaves the cell, gets neutralized, that might be actually also something really promising. Right now, no, there's no cure, there's no therapy out there that helps.
Is there any information on whether any of the tauopathies are hereditary or familial?
Yes, I see where you're coming from, and psychologically, or when I'm on the phone, I try to avoid this topic, because there is a very, very small fraction of people and families in the US where it runs in families. My problem is, and now, because you can Google that, you can read of that, and then my problem is, the moment I say this, you get up, you leave today, and you're worried that it runs in your family, because Alex said, well, hey, it can be. He didn't say no. So I think if you go home with a no, it's not familial, then I think that's fine. It's not the full truth. Yes, there are families, and particularly Dr. Bouvet actually runs a trial in a study where he looks actually at, and he will talk about this later, about the familial cases of PSP and related diseases. Absolutely, yes, and they are of great interest for the research community, because they might give us hints, of risk factors. What is actually going wrong? Can we actually use those families and study what's going on for future drug targets, for example? So don't worry if your close family members, if they are not affected, then you're fine. But it's just bringing the message across. It's a non-hereditary disease. It's only, I think, 4%, or is that right, Brad? Yeah, it's very small. There's another question. Oh, sorry, here. Right.
Thank you for what you do. Earlier you said PSP and Alzheimer's share the same misfolded tau. How come Alzheimer's drugs, because there are some, don't work for PSP people?
They right now do not address tau. So what Alzheimer's drugs do right now is they look at different areas. And then in the past also, there is another protein called beta amyloid. Not that they're working right now, but this is where the Alzheimer's field really was focusing on. So, and also the drugs you use in Alzheimer's, they give you an extended span of good quality of life, but they really do not modify the disease in itself, right? You still have the progression. It slows it down, but there is certainly no cure. And again, they do not focus on tau. And the shared part between the two diseases is actually tau. Yeah. So one last question? Okay. No. Well, you can find us later on during the breaks, and I'm happy to talk to you. And thank you very much again. Also, thank you, Sarah.
Thank you.
And